Our next presenter is Damir Creasy, also from Villanova University. And based on Nicole's introduction, unofficial introduction, Damir has some great findings to share with us.
Okay. How's it going, everybody? Today I'm going to tell you about an experiment I performed to investigate mangrove tolerances to inundation using elevation as a proxy. Understanding mangrove survivorship in relation to sea level is an important goal for wetland scientists. So to study this, I employed a novel approach to traditional marsh plant elevational studies. And I'm going to go over that methodology and my results with you today. So where I performed my experiment was the North Matanzas field site. It's been introduced by Anna already, so I'm not going to dive too deep into it, but some important things to know about this site is that the average elevation is around 48 centimeters. And above sea level, by the way. And this is measured in NAVD 88. The dominant vegetation at this site is Baetis maritima, Spartina alterniflora, Barrickia frutescens, and Avicennia germinans. So before I get into my study, a question I want to ask is, as mangroves migrate poleward, is it possible to predict where they'll end up using what we know about elevation? And with that, I want to present a study by Leong et al. 2018 that shows a zonation pattern along surface elevation. So this y-axis is measured in meters above sea level. And as you can see here, there is a clear zonation pattern between all these species of mangrove. The mangroves that I will be focusing on are the Avicennia species, specifically, or Avicennia genus, specifically the black mangrove. And if you look here, these red boxes highlight that there is a zonation pattern within Avicennia genus. So getting into my study, why focus on elevation? And that is because elevation is tightly linked to inundation. The higher places tend to have less inundation and lower places tend to have more inundation. This changes based on, sort of barriers and human built things. But for the most part, in an unimpeded wetland, higher elevations are going to have more inundation and less inundation, and lower elevations are going to have more inundation. The NMAT surface elevation range is actually negative 20 centimeters to around 95 centimeters above sea level. So getting into my questions, I wanted to ask, what is the survival rate of Avicennia germinans at different elevations in the salt marsh mangrove ecotone? My hypotheses I came up with for this question were that the survivorship of Avicennia germinans is closely linked to elevation, as well as elevation will have a dramatic effect on the zonation pattern of Avicennia germinans. So for my experimental design, this is where I get into the novel part of it. My experimental design is focused around a proposed ideal elevation range for Avicennia. This is based on field surveys done by William Vervaeke. He, sort of went around and measured elevations of different plants, and we sort of saw that the mangroves sort of fell mainly between 30 and 70 centimeters above sea level. So my experimental range was to encapsulate this whole proposed range, and I planted a bunch of plants along this range to get at the survivorship of these plants as the elevation changes in gradient. So how I executed the experiment. First, we did a field survey of the NMAT site to find proper sites. And if you look here, you can see these berms along this this creek. This is at the NMAT site. And these berms are, sort of artifacts from construction of this drainage ditch, this drainage ditch, which created this perfect elevational gradient that I was able to plant my plants along. So if you look here, there's about a 20 centimeter difference between each elevational treatment. I planted my plants in triplicate at each elevational treatment. And this is just a picture of me planting my plants. So for my results, what I found out is that there is an extremely tight link between elevation and survivorship, as shown by this logistic regression here. One thing to note about this logistic regression is that the y-axis here is actually in relation to the curve that you see. It's not in relation to the verticality of the points. The points only relate to the elevation that you see. So at, around zero elevation, elevation, there's one plant that survived, and there are a bunch of plants that died. So diving into that, or getting more into that, you can see I have a very, very significant p-value here. Anyway, but getting into that and sort of diving deeper into that, I have these three graphs here that show average survivorship on the y-axis here, the average height on this axis here, and the average amount of leaves here. This is how I measured growth rate over time. As you see, the growth rate changes as time goes. And there's a couple weird things that I wanted to show here. Before I get into that though, the average survivorship sort of followed this whole logistic regression here. As you went up in elevation, there was a increase in survivorship. So the average heights, this was a very weird thing that you'll see. The average heights are actually flipped in relation to this average survivorship. So as you went lower in elevation, the average height actually increased. What I believe that is a survivorship bias. So the plants that were able to stick their heads above water essentially were able to survive at these higher inundated sites that are, there's a bit more pressure there. The average amount of leaves kind of went a little wonky, but it sort of changed over time. Anyway, going on. So there were some other factors that I measured that had an effect on survivorship, or showed an effect on survivorship. I averaged survivorship within my plots, so that's why you're seeing these like little four levels of survivorship. This is the average between 3 plants, so it's between 0 and 100% survivorship. But I did see a significance between redox and survivorship and plant cover, the percent plant cover and survivorship. So in order to tease these factors apart, what I did was regress redox and plant cover against elevation. And what it seems is that redox and percent plant cover is an artifact of elevation. So essentially, elevation is a proxy for redox in this measurement. So what I did, I performed a factorial ANOVA to sort of see which one of these factors was affecting survivorship the most. And within that model, I found that elevation had the strongest effect, along with elevation cross with redox potential. Redox potential was measured to 2 centimeters within the soil. And it's sort of, I still believe, I believe this shows that it's a symptom of elevation essentially. So to sort of tease apart plant cover as well, I saw the same thing. plant cover was a symptom of elevation, essentially. Specifically, the types of plants, as you see here on the right, the types of plants change as elevation changes. And there is sort of a competition effect. Over here, you can see that with Batis going down and Salicornia going up, actually, this little fun effect. But I crossed all these things with elevation, or I ran an ANOVA of all these things against elevation, and they were all significant. All of the amount of plant cover was significant across elevation. So that to me signifies that elevation has a strong control over the zonation pattern and percent cover of plants in the of other marsh plants. So to investigate my second question, this second question is an exploratory question, and it's sort of an application of what my results from this study. And the question is, can I use the survival data I collect to create effective habitat models, which will help determine the establishment locations of future Avicennia germinans and bolster our knowledge of marsh equilibrium? So I don't present a hypothesis for this question, but because it's exploratory. So going on from that, yes, you can. And here is the habitat suitability model that I created using my data. The way that I did it, I took a digital elevation model and I transformed that with math and a little bit of insight from my experiment, and spat out a habitat suitability model. And this has a lot of restoration implications, as I sort of, hinted at. But those implications include informing restoration efforts and planting. So what you see here, this habitat suitability model shows you where mangroves will most likely survive. Okay, so the greener areas have higher likelihoods of survivorship. So if you were to, perform some sort of restoration or planting, you would most likely do it, or you would have the best results within these more green areas. Some other implications include understanding where mangroves will move northward and fill in to dominate ecotonal wetlands. So we can make the assumption that the mangroves will end up in these more suitable areas because that's where they'll survive. And we can also assess areas at high and low risk of inundation just by tweaking this model a little bit. This model, it using the same sort of math, you can just apply that to inundation that you want to see, or like critical inundation rates, and sort of understand
3
the same
minutes.
thing. So with that is my my whole presentation, and I'm free for questions.
Great. Thank you so much, Damir. That was a wonderful presentation. We had a few questions come into the chat box. Do you think your suitability model can be adapted to other areas?
Absolutely. So the whole point of this, or the suitability model, all it takes to adapt to another area is just having the data for that area. If you have LiDAR data or a digital elevation model, it's a very simple thing to just apply, sort of transform that into whatever purpose you need. So it is very translatable. And in fact, my whole experiment was to translate this into more further up and more northern sites, just to sort of see if we can predict. And it's very possible.
Awesome, thank you. There was a comment that mangroves at the leading edge maybe are more like shrubs. compared to the mangroves at the southern trailing edge that grow more like trees. Do you think that that relationship might be tied to what you found in the response to elevation?
I don't think so. This study was performed on yearlings and saplings, and sort of wanted to get at that, I guess, R response, of mangroves being an R species, and that sort of critical first year of them surviving. I don't think that it is a artifact of elevation. So I think that answers your question.
Have you seen whether the LiDAR data agree well with the ground survey data that you collected?
Yeah, absolutely. So this is all LiDAR data, and the LiDAR data essentially, it's there are some problems with LiDAR, especially in wetlands, specifically being that there's plant cover that may impact where it's measuring surface elevation, as well as refractions from water. But from what I saw, this was the LiDAR data that I used was a flyover from the Hurricane Irma. And what I did was I sort of checked to see if that data was accurate with my field surveys when I was going through and searching for locations, and it seemed to be very accurate. Yes.
Based on the results from the model that you created, have you done any ground truthing to determine if there are mangroves in the areas that you predicted to be suitable habitat for them?
I haven't done any ground truthing, just a little bit of remote sensing. And you do sort of see a relation, but as all models are useful, all models are wrong, some are useful, and that's sort of the thing I wanted to, that's sort of in the back of my head whenever I use a model for something.
Thank you. Have you considered adding some sort of proxy for recruitment potential to the model? There's a comment that maybe some anthropogenic barriers such as seawalls or distance from known seed sources could maybe be a consideration to think about.
Yeah, absolutely. That's a huge something huge I need to sort of hone in on. I haven't added anything into the model, but it is definitely a factor that can be added in, and I think it would be pretty easy to implement as well.
Great. Sounds like you have some future work as well.
Yeah, absolutely.